Picture this. Two animals are standing 20 meters apart in a forest clearing. They are, by every genetic measure available to modern science, essentially the same creature. Same species, origin, same skeletal architecture, same basic organ systems, same ancestral behavioral repertoire. If you extracted DNA from both of them and handed the samples to a geneticist without labels, the differences would be subtle enough that distinguishing them would require careful, targeted analysis rather than an obvious read. One of them is a wolf. It is watching you with the particular quality of attention that large predators have: total, calculating, completely unreadable. It is deciding something. You do not know what. The distance between you and it feels insufficient in a way that your nervous system is communicating very clearly and your brain is not in a position to argue with. The other one is a golden retriever named Biscuit. Biscuit has just noticed you and is now approaching at a pace that can only be described as enthusiastic structural failure. All four legs operating with maximum commitment and minimum coordination, tail moving at a frequency that should probably concern a physicist, expression communicating something in the neighborhood of the greatest happiness any living organism has ever experienced at the sight of another living organism. Same animal. Profoundly, almost incomprehensibly different creatures. And the question of what actually separates them, not superficially, not just in terms of behavior or appearance, but at the deep biological, neurological, developmental, and evolutionary level, is one of the most fascinating questions in the entire study of how ancient humans reshaped the natural world. Because the answer is not simply that one was bred to be friendly and one wasn't. The answer involves developmental biology, neuroscience, a Soviet Fox experiment that sounds like it should be fictional, the concept of domestication syndrome, and the startling realization that the gap between a wolf and a dog reveals something fundamental about how radical biological transformation can happen far faster than anyone previously believed possible. This one goes deep. Stay with it. Start with what they share because the shared foundation is what makes the differences so remarkable. Dogs and wolves are classified as Canis lupus, the same species, with domestic dogs designated as the subspecies Canis lupus familiaris. They share approximately 99.9% of their DNA. They can interbreed and produce fertile offspring. Their basic skeletal structure, their organ systems, their sensory capabilities, their fundamental behavioral repertoire, all of these are recognizably similar to anyone who spends time with both animals. A wolf howls. A dog howls. A wolf forms social bonds within a pack structure. A dog forms social bonds within whatever social group it finds itself in. A wolf uses scent, sound, and body language to communicate. So does a dog. The shared foundation is real and deep, which is precisely why the differences are so extraordinary to examine. Because starting from that shared foundation, thousands of years of domestication have produced changes in dogs that extend from the obvious and visible all the way down to the neurological and hormonal, changes so fundamental that some researchers argue the domestic dog is, in meaningful developmental terms, a different kind of organism than a wolf, even if the genetic distance between them is smaller than the genetic distance between some subspecies of birds that nobody would hesitate to call the same animal. Begin with the most studied and most surprising difference: the social cognition gap. This is where the dog-wolf separation becomes most dramatic and most relevant to understanding what ancient humans were actually dealing with when the domestication process began. Dogs are extraordinarily, almost unnervingly good at reading human social signals. A dog can follow a human pointing gesture, look in the direction a person is pointing to find a hidden object, with a reliability and ease that chimpanzees, our closest living relatives with vastly larger brains relative to dogs, struggle to match. Dogs track human eye gaze. They respond to human emotional expressions. They show what researchers describe as referential looking. When confronted with a problem they cannot solve, dogs look back at nearby humans in a way that wolves, raised identically from birth alongside humans, do not consistently do. This last point deserves emphasis because it eliminates the obvious explanation. The social cognition difference between dogs and wolves is not a product of experience or learning alone. Wolf pups raised by humans from birth, socialized intensively, given every opportunity to learn human social signals through direct experience. These animals still show significantly weaker performance on tasks measuring human social signal reading than domestic dogs. The difference is in the biology. Dogs are born with neural architecture that is specifically tuned to human social signals in a way that wolves simply are not, and no amount of early socialization produces that tuning in a wolf. Where did this neural architecture come from? This is where the domestication process itself becomes fascinating, because the cognitive changes in dogs were almost certainly not directly selected for in any deliberate sense. Ancient humans were not sitting down and deciding that they wanted an animal specifically optimized to read pointing gestures. They were selecting, initially and probably unconsciously, for reduced fear responses toward humans and reduced aggression in human proximity. And those behavioral changes dragged along neurological changes as a consequence of the developmental biology they are embedded in. The mechanism connecting behavioral selection to broad physical and neurological change is called domestication syndrome, and understanding it requires a brief excursion into developmental biology that is worth every second of the detour. In vertebrate embryos, a population of cells called neural crest cells migrate from the developing neural tube to destinations throughout the body, where they differentiate into an extraordinary range of cell types: adrenal gland cells, pigment cells, cartilage, and bone cells in the face and skull, and neurons in parts of the peripheral nervous system. The neural crest is one of the most remarkable developmental structures in vertebrate biology, and it is exquisitely sensitive to developmental timing. Animals selected for reduced fear and aggression toward humans show reduced reactivity of the hypothalamic pituitary adrenal axis, the stress response system, which appears to correlate with subtly altered neural crest cell migration and differentiation. And because neural crest cells contribute to so many different structures, a change in their developmental dynamics produces changes across a whole suite of seemingly unrelated physical characteristics simultaneously. Floppy ears, because the cartilage-forming ear structure comes from neural crest cells and reduced developmental input produces softer, less upright ear cartilage. Shorter, more rounded snouts, because facial bone and cartilage are neural crest derivatives. Smaller teeth. Piebald, or patchy coloring, because pigment cells are neural crest derivatives. And reduced neural crest migration means reduced pigmentation coverage. Curled tails. Retention of juvenile physical features into adulthood, a phenomenon called neoteny. Smaller brain volumes relative to body size than wild counterparts. None of these features were directly selected for in the initial domestication process. They came along for the ride, biologically linked to the behavioral traits that were selected, because the developmental system that produces them is the same developmental system that produces the reduced stress reactivity that humans were selecting for. The package deal nature of domestication syndrome is one of the most striking discoveries in the biology of domestication, and it explains why domestic animals across wildly different species, dogs, cattle, pigs, horses, sheep, even domestic foxes, show overlapping suites of physical changes that parallel each other even though they were domesticated separately. Which brings up the Soviet fox experiment, because it is impossible to discuss domestication syndrome properly without it, and because it is the most direct experimental demonstration of how fast domestication-related changes can appear, and how completely they recapitulate the dog-wolf transition in an accelerated timeline. Beginning in 1959, Soviet geneticist Dmitry Belyaev began a selective breeding program with wild silver foxes, selecting exclusively for reduced fear and aggression toward humans. The same behavioral traits that appear to we have initiated dog domestication. No selection for physical appearance, no selection for cognitive traits, just behavioral tameness. Within approximately 10 generations, a span that is geological eye blink in evolutionary terms, the selected fox population began showing physical changes that nobody had directly selected for. Floppy ears, piebald coloring, curled tails, shorter, rounder snouts, retention of juvenile vocalizations and behaviors into adulthood, changes in reproductive cycling, the domestication syndrome package appearing spontaneously as a consequence of selecting for tameness alone, within a time frame short enough that the researchers who initiated the experiment lived to observe it. The experiment, which has continued across decades and multiple generations of researchers, demonstrated several things simultaneously. First, that the physical changes associated with domestication are developmentally linked to behavioral changes in exactly the way the neural crest hypothesis predicts. Second, that these changes can appear remarkably fast under strong selective pressure, far faster than previously assumed. Third, that the dog-wolf transition, which might seem to require vast timescales to produce such dramatic differences, is actually achievable within a relatively small number of generations when the selection pressure is consistent and strong. For ancient humans, this has an important implication. The initial domestication of wolves into proto-dogs did not require thousands of years of deliberate breeding programs. The early stages of the transition, from wary, human-avoiding wolves to tolerant, human-associating proto-dogs, could have happened within a human lifetime or a few generations, fast enough that the people involved in the process would have witnessed meaningful change. The later stages, the full elaboration of dog-specific cognitive and social traits, took longer. But the process could have been initiated, sustained, and yielding observable results within timeframes that ancient humans could actually perceive and respond to. Now move to the neurological differences between adult dogs and wolves, because this is where the gap between the two animals becomes most concrete and most surprising. Studies examining brain structure and function in dogs versus wolves have identified differences in several regions, including areas associated with fear processing, social behavior, and reward responses to human interaction. Dog brains show reduced amygdala reactivity to novel stimuli compared to wolves, reduced fear response to unfamiliar things, which correlates directly with the behavioral tameness that domestication selected for. Dog brains also show stronger activation in reward-related neural circuits in response to human social cues. The presence of a familiar human eye contact, human voice, than wolf brains do. This neurological difference has a hormonal dimension that is particularly striking. When dogs make eye contact with their owners, both the dog and the human show increases in oxytocin, the neurohormone associated with social bonding, trust, and affiliation. The same effect has been documented in parent-infant interactions in humans. The bond between a dog and its owner, at the neurohormonal level, appears to engage some of the same biological machinery as the bond between a parent and a child. Wolves, even hand-raised wolves with extensive human socialization, do not show the same oxytocin response to human eye contact that dogs do. This is not a small or superficial difference. It suggests that the domestication process reshaped at the neurohormonal level the way dogs process and respond to human social interaction. That dogs are not wolves who have learned to tolerate humans, but animals whose social bonding neurobiology has been partially redirected toward humans as a primary attachment target. The redirection appears to be developmental, embedded in the biology rather than learned through experience, which is why even puppies with minimal human socialization show stronger human-directed social behaviors than intensively socialized wolf pups. The developmental timeline differences between dogs and wolves add another layer to this picture. Wolf pups have a socialization window, the period during early development when they most readily form social bonds with other species, that opens earlier and closes faster than the equivalent window. In dogs, wolf pups that are not exposed to humans before approximately 3 weeks of age become increasingly difficult to socialize toward humans. By 14 weeks, the window has largely closed. Dog puppies have a longer and more flexible socialization window, allowing social bond formation with humans across a wider developmental period and under a broader range of conditions. This difference in socialization window timing has enormous practical implications for the domestication relationship. A dog puppy that arrives in a human home at 8 weeks of age is still well within its socialization window and will form robust social bonds with the humans it encounters during that period. The same timing for a wolf pup would be late in its socialization window, producing a less complete and less stable social bond even with intensive effort. The domestic dog, in other words, has been shaped not just to be willing to bond with humans, but to be developmentally primed for that bond across a timeline that makes the bonding practically achievable in normal human-dog interaction contexts. Physical differences between dogs and wolves extend beyond the domestication syndrome features already discussed into areas of metabolism and dietary adaptation that are directly relevant to how and why the domestication relationship became sustainable. Dogs show genetic adaptations for starch digestion that wolves lack. Multiple copies of the AMY2B gene, which codes for salivary amylase and allows more efficient processing of starch containing foods. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Wolves, as obligate carnivores in their natural ecology, have little evolutionary pressure to develop starch digestion capacity. Dogs, living in proximity to humans who were increasingly cultivating and consuming grain-based foods, were selected for the metabolic flexibility to thrive on a more varied diet, including significant carbohydrate content. This dietary adaptation is not a trivial accommodation. It represents a fundamental metabolic shift that made dogs viable as human camp followers and household animals in a way that wolves, metabolically, are not well suited to be. A wolf fed a human-style diet performs poorly compared to one on its natural high-protein diet. A dog is metabolically equipped to handle the dietary diversity of human food environments in ways that reflect genuine adaptation to the human ecological niche rather than simple behavioral accommodation. Morphological diversity in domestic dogs, the extraordinary range of body sizes, skull shapes, leg lengths, coat types, and overall physical forms present across the 300+ recognized breeds, has no parallel in wolf populations, and represents one of the most extreme examples of within-species morphological variation in the animal kingdom. The genetic basis of this diversity has been partially mapped, and what the mapping reveals is striking. A relatively small number of genetic variants, each with large effects on specific physical traits, account for most of the morphological diversity across dog breeds. The genetics of dog diversity are, in some respects, simpler than expected. Not thousands of small effect variants producing gradual change, but a smaller number of high effect variants that can produce dramatic physical differences when present or absent. This genetic architecture of diversity is itself a product of domestication, the result of repeated population bottlenecks, founder effects, and intense selective breeding within breed lineages that reduced genetic diversity within breeds while concentrating trait-determining variants. Wild wolf populations show far more genetic diversity within a population than most dog breeds do, but far less morphological diversity between populations than exists between dog breeds. Domestication compressed genetic diversity while amplifying morphological range, producing the paradoxical situation where a species with reduced overall genetic diversity shows more physical variation than almost any other comparable group of animals on Earth. For ancient humans, the initial stages of this process were probably invisible as deliberate morphological shaping. The first ancient humans who began associating with proto-dogs were not trying to produce a greyhound or a pug. They were observing that certain animals were useful, for alerting the camp to approaching predators, for assistance in tracking and hunting, possibly for warmth, possibly for the same reasons. Humans have always been drawn to animals that respond to them positively. The physical shaping came later, as the relationship deepened and as humans began actively selecting for specific functional and eventually aesthetic traits. But the social and neurological shaping, the redirection of canine social bonding toward humans, the amplification of human social signal reading, the reduction of fear and aggression in human proximity, this appears to have been the foundation on which everything else was built. Ancient humans did not domesticate wolves by selecting for floppy ears or short snouts. They domesticated wolves by initiating and sustaining a relationship with the subset of the wolf population that could tolerate, and then seek, and then prefer human proximity. Everything else followed from that initial behavioral selection, carried along by the developmental biology that links behavioral traits to physical ones in ways that ancient humans could not have understood, but absolutely benefited from. The wolf and the dog are separated ultimately not by the vast genetic distance that their extraordinary behavioral and physical differences might suggest, but by the accumulated consequence of thousands of years of selection pressure operating on a tight developmental system. A small shift in neural crest cell dynamics, driven by selection for reduced stress reactivity, cascaded through the developing organism to produce floppy ears and piebald coats and rounded snouts and smaller brains, and longer socialization windows and redirected oxytocin responses, and starch digesting enzymes, and the ability of Biscuit the Golden Retriever to communicate with his entire body that seeing you is the best thing that has happened to him today and possibly ever. The wolf in the clearing is extraordinary. Watching it, you are looking at an animal shaped by millions of years of natural selection into one of the most successful predators on Earth. Efficient, socially sophisticated, ecologically flexible, built by the most rigorous testing process the natural world provides. There is nothing deficient about a wolf. It is exactly what it should be. The dog is also extraordinary, for entirely different reasons. It is an animal that has been reshaped at the neurological and developmental and metabolic level by its relationship with a single other species, reshaped so completely that it now forms social bonds across a species boundary with the ease and depth that most animals reserve for members of their own kind. That is not a diminishment of the wolf form. It is a different kind of achievement, a testament to how profoundly and rapidly a sustained selective relationship can transform a living organism. Ancient humans who first began that relationship did not know what they were starting. They could not have imagined Biscuit. They were solving an immediate problem, or perhaps not even solving a problem, but simply tolerating and then enjoying and then cultivating the presence of animals that made their lives slightly less difficult in a world that was actively trying to kill them. Across thousands of years, that initial tolerance built the most enduring and most intimate interspecies relationship in human history. The wolf became the dog, and in doing so, it became something that the wolf, for all its magnificence, has never been and was never trying to be, a creature whose entire social world is organized around its relationship with us.